Hello and welcome to Biology 151 at Marymount University. So as you know, this is going to be a flipped class, and so the lectures will be recorded in this fashion. You will have access to all of the PowerPoints, and you should be checking your prep sheet and answering the questions on your prep sheet as you watch the video and go through the PowerPoints. And then we will be doing activities in class to support the information that you are learning as you watch this lecture. So we're going to start out, chapter 1 is going to kind of go through some of the major themes, the major ideas that we're going to talk more about as we go through the rest of the semester. But chapter 1 is really just a nice introduction to this class and to the rest of your textbooks. We'll talk a little bit about evolution, and we're talking about scientific inquiry, which you guys probably already know a good bit about, and just some of the major themes in biology. So the first question that we need to kind of talk about is, what is the definition of biology? And we have a pretty good definition, is the scientific study of life. So we know that biologists ask questions, and we strive as biologists to answer those questions through our research. And the good thing about biology is it's broad enough that we can ask a lot of different types of questions. We can work on a scale of cellular biology and think about how does a single cell work and how does it interact with other cells and how does it develop into an organism. We can kind of take the middle of the line approach, thinking about how does the human brain work, so it's part of an organism, or all the way up to sort of a macro scale in biology where we think about how living things interact in a community. So biology is a very broad topic that talks about or that thinks about life in a very large way, from subcellular to all the way up to populations and communities interacting. So what I'd like for you to think about, and we'll talk about more in class, is a one-sentence definition of life. What are some things that you would use? How would you describe life in one sentence or less? So I will be asking you to do this when we get to class. So be thinking about that as you prepare for class and as you go through the rest of this talk. So we actually have some things that do define life. There are some characteristics of life that, as biologists, we have said that all living organisms must have these specific characteristics. So first of the first one of those that we want to talk about is order. So as biologists, we like to categorize and order things, and all living things must fit into our categories. So we classify and we distinguish living things in a certain way that we relate them to other living things. The second characteristic that we like to talk about is evolutionary adaptation. So this means that organisms have evolved, living organisms have evolved over time, and we'll talk more about that later on in this talk. But we know that through the process of evolution, organisms have adapted to their environment. So related to that is sort of the next characteristic, and that is response to the environment. Living organisms have to be able to respond to the conditions in which they are living. So a good example of this might be sweating. So we're sitting on the beach, it's a hot day, and our body starts to sweat. So that's one way that our body responds to its environment, is by cooling off our body temperature by sweating. Another example may be the long neck of a giraffe. So it could be that at one point giraffes were living in a community where all the trees were really high and all the leaves were at the top of the tree, and so the giraffes with the longest neck were able to reach the leaves and eat and survive, and that is the trait that was passed down from one generation to the next. And we'll talk more about that. I'll give you an example of that in terms of bacteria in a moment. The other thing that all organisms do, and we'll kind of lump these two together, is they reproduce and they grow and develop. So all living organisms do these things, from a single-celled amoeba all the way up to humans. Every living organism along that spectrum grows and develops, and it reproduces offspring. The other thing that organisms have to do that's really important, and we'll spend a whole couple of chapters talking about this, is they have to process energy. We have to be able to obtain energy from our environment and convert that energy to a form that we can use. So now you all know the law of conservation of energy that says energy cannot be created or destroyed, but we do have to be able to convert energy into a form in which we can use it, and all living organisms have to do that. The last thing, and this kind of goes with that response to environment cue, is that all living organisms have to have a way to regulate their status quo. So we call that homeostasis. That is sort of the natural conditions in which that organism survives and thrives, and all organisms have to have a way to regulate that. So we'll talk more about all of these things as we go through the rest of these chapters, but this is just kind of a nice introduction to some characteristics that are associated with living organisms. Okay, so we've touched on this a little bit in terms of the levels of biological organization. So we know that life can be studied at all different levels, from molecules all the way up to the living planet. So we can go from a very small microscopic view of life all the way up to very large sort of macroscopic view of life. So this is an enormous range of information and a tremendous quantity of living organisms that we have to classify and organize. And so that led to some pretty sophisticated organizational systems or organizational methods. You guys have probably all heard of the king, phylum, order, that long thing that people forget and don't really remember. And it is still used, and you'll talk more about that when you get to bio152. We're not going to spend a lot of time in this class talking about that type of classification, but there is a way that we like to classify living things, and it fits very nicely with what we do in bio151, and that's what I want to talk to you about next. So we first start, this is our sort of biological organization chart that we'll use throughout this class period. We start down here throughout this class. We start down here with atoms. So we know you probably think of the periodic table when you think of atoms, right? These are things like your carbon, your hydrogen, your oxygen, your nitrogen, your sulfur. You guys have all seen a periodic table before, and that's where we start in this class. Now, atoms, remember, are not living. They do not display those characteristics of living things that we talked about. So atoms are not living, but you're going to see how they fit into the organization of living things as we go through this chart. The next sort of hierarchy level on our chart are molecules. So molecules are made up of atoms. Now we talk about 4 really important molecules in biology. So these are called the molecules of life. So we have carbohydrates, lipids, proteins, and nucleic acids. We're going to spend a chapter talking just about these 4 types of molecules. So again, these molecules themselves are not living, but they are called the molecules of life because they make up life. That brings me to our next level here, which is the cell. And our cell is the smallest unit of life that we talk about. So the smallest living thing that we talk about is a single cell. Remember, atoms and molecules are not living, but they make up cells, which are our smallest unit of life. So that's the first level where we see life on our hierarchy system. We know that cells make up organisms. So that's our next level. We can have single-celled organisms such as bacteria, or we can have multi-celled organisms such as yourself. So you can see the range of organisms that we have that fit into life, that are made up of cells, that we have to sort of classify and think about in terms of from a biological perspective. From organisms, we'll pick it up here with populations. So when we talk about populations, we talk about organisms of the same species or kind living in an area. So for example, in this picture here, we have a population of zebras, we have a population of trees, and we have a population of grasses. So these are three separate species all living in the same area. They're organisms. The zebras are all organisms. The trees are all organisms. The grasses are all organisms. But we group them together with their like kind, and we call that a population. From there, our next level in our hierarchy is the community. So the community is all of these populations taken together make up the community. So when we take everything that's in that wooded area, all the zebras, all the trees, all the grasses, all of those things together make up the living community. And they all affect, all of the organisms are affected by the other organisms in that community. If all the grass were to die out, that would affect the zebras. If all the trees were to die and topple over, that would affect the zebras as well as the grass. So all of these things in a community are interconnected and are affected by others. The next level in our hierarchy is the ecosystem. So the ecosystem is the community, so everything that makes up the community, plus the physical environment. So we're back to sort of non-living things here, right? We're talking about soil, water, and rocks. All of those things are non-living, but they affect the living organisms in that community. If, let's say in our zebra example here, there's a little stream or a little creek running through where these zebras are living. If that stream were to dry up, that would affect the living organisms in that community. The grass might die, the zebras might have to move so that they could find water. So even non-living things within our environment affect the living organisms. And then lastly, we talk about the biosphere. So everything present in the Earth's crust, also in the water and in the atmosphere. All of those things also affect the living organisms, where they live, how well they thrive. So that's sort of our highest level of hierarchy when we talk about biological organization and living things. The other way that we classify things in bacteria, in biology, is based on the type of cells that make up the living organisms. So if we start down here, we're going to talk about this in terms of origin of life. We're going to talk about three main categories or three main buckets, so to speak, where we can classify organisms. So the first one that I want to talk about is the bacteria. These are our one-celled, so single cellular organism, and they are prokaryotes. So prokaryotes are cells with no nucleus. So pro actually means before, karyote means nucleus. So these are organisms that evolved before nuclei. So these are only prokaryotic cells. We know bacteria to be very widespread. They grow in lots of conditions. They have great metabolic diversity, meaning that they can grow in almost any environment. Even though these are simple organisms, simple cell, one cell prokaryotic cells, about the simplest cell that we can have, they are still quite complex because of this metabolic diversity and the how widespread they are. The second type of organisms that we talk about are the archaea. So the archaea are also prokaryotes. So again, one-celled prokaryotes. But they have, the difference is archaea can grow in very extreme habitats. So places where we don't see bacteria. So for example, the frozen tundra of the Antarctic, or the hot springs, boiling hot springs. We wouldn't see bacteria able to survive in those types of environments, but we do see archaea that can grow in those types of environments. So again, they are one-celled prokaryotes, very simple organisms, about as simple as we can get, but they can grow in these very extreme environments. The third and most complex of the categories that we talk about are the eukarya. So eukarya are our eukaryotes. So these are cells that do contain a nucleus. So that prefix means true, and again, karyo is nucleus. So these are cells that do contain a nucleus. So they're more complex cells than the prokaryotes. And organisms that fit into the eukarya category can be either single-celled or they can be multicellular. So this is a wide range, very diverse range of organisms here. So you can have your protist fit into this category. So these are, they can be one-celled all the way up to, the giant seaweed. So these can be kind of a wide range of organisms here. They are what we call producers, so they can either produce their own food through photosynthesis. They are consumers, so they can consume other organisms. They are also decomposers, which means that they can survive on other nutrients in their environment and decompose those nutrients in order to sustain themselves and get their energy. The second category that we want to talk about, kind of a subcategory of eukarya, are the plants. So we know that these are organisms that carry out photosynthesis, which makes them producers by nature. The definition of a producer is an organism that can produce its own food, and that's what plants do through the process of photosynthesis. The third subcategory of eukarya are the fungi. So again, these can be one-celled or multicellular. These are mostly decomposers, so they sort of digest nutrients in the environment, and that's the way they obtain their energy. And they digest usually externally and absorb externally, The last subcategory of eukarya are the animals. So animals we know are multicellular. They have the capacity to move about, which a lot of the other organisms in the other three subcategories do not necessarily. And they are animals, as we know, are consumers. So we consume either other producers or other consumers in order to obtain our energy. So that's what makes us consumers. Okay, don't quite know why this slide is a little crooked, but in terms of this diversity, and we've talked about the fact that organisms have we have this very wide range of organisms in biology, from single-celled bacteria all the way up to animals. How did we get there? How did we get all of this diversity in organisms? And it really comes into play here when we talk about evolution and natural selection. So the definition of evolution is when various forms of a trait either increase or decrease in a population over time, over successive generations. So what are the traits that are passed down or eliminated as a population procreates and reproduces? So we know that this happens through a process of natural selection. So we talk about natural selection. We know that members of a population vary in traits, and then they pass those traits down to the offspring. We know that certain forms of a trait can be better adapted to an environment than others. So we used the example earlier of the giraffes with long necks. So let's say, for example, there were two types of giraffes, and there were short-necked giraffes and long-necked giraffes, and all the trees were really high and all the leaves are way up high. Well, is the long-necked giraffes going to be able to reach those leaves and be healthy and survive and reproduce and pass that trait of having a long neck down to their offspring? The short-necked giraffes aren't able to reach the leaves and eat the leaves. They're not going to be as healthy. They're not going to be reproducing, and eventually that trait is going to we're going to see that trait less and less often in the population until it is no longer there. So we know that different traits are better adapted to the environment, and individuals with the better adapted traits are more likely to survive and reproduce than those without the traits. So the long-necked giraffes are more likely to survive and reproduce than the short-necked giraffes. So that trait is going to be better adapted to the environment, and eventually that population is going to evolve. This can lead to new species if the population becomes very different from the rest of the original members. So let's talk about this in terms of bacteria. So again, on the left-hand side over here, we sort of have that process of evolution that we just talked about. And on the right side, I'm going to show you, give you this in an example. So what we have is there is a population of bacteria that vary in their resistance to penicillin. So this is an adaptive trait. So bacteria that are resistant to penicillin will survive better in a penicillin environment. If we put these bacteria on a plate that contains penicillin, we would expect the bacteria that are resistant to it to be able to grow. Those that are not resistant to penicillin will not grow. So we take, what we have here is, we have our plate and we have penicillin. So I'm going to put plus pen here because we have penicillin on this plate. And then we have several different types of bacteria. The gray ones have no resistance to penicillin. The pink ones have a slight resistant. The light pink, the dark pink are more resistant, and the red are our most resistant. So we would expect, as this population grows, we would expect those red and probably those dark pink bacteria to survive, to flourish. We would expect the ones that have either no or slight resistance to penicillin to die off, to not be able to live as long and not be able to reproduce. So over time, our population of bacteria would change and we would end up with all hot pink and red bacteria, and these guys, the gray ones and the light pink ones, would be eliminated from the environment. That is the process of evolution. So you can see that's exactly what happened here. As we let these bacteria grow and reproduce and grow and reproduce over time, we ended up with the hot pink guys and the red guys surviving and lasting, and the top two types of bacteria were no longer able to survive in that penicillin environment. So that is an example of how evolution works to change the population of an organism over time. All right, so I want to end this a little bit talking about the scientific inquiry process. So again, I think this is a process with which you are all familiar. You've all heard about the scientific method, and you've talked about the steps of that method probably since, I don't know, 5th or 6th grade biology. So you're all familiar with that. I'm not going to go through that as much. The key that what we want to remember is that our science needs to be hypothesis based. We always need to be thinking about designing experiments and doing research based on a hypothesis. However, when we think about the scientific method, we normally think about it as a starting point. We would have a starting point with our question, and we would follow a unilateral path to a conclusion. And what I want to propose today is that that is rarely the way science happens. More often than not, we do a lot of backtracking and a lot of rethinking as we do biology and as we do any scientific research. And so more often than not, our methods look a little more like this, as opposed to that unilateral, unidirectional approach that we often are taught of how the scientific method works. So yes, most of the time we are starting out with forming and testing hypotheses. So we need to sort of think about what it is we want to explore and discover, and then we want to come up with a testable hypothesis for that. We might do community analysis or get feedback from our community. We might think about the benefits of this research and how it's going to affect society and what the outcomes are going to be. And that might lead us to new and different questions that we need to explore and discover. And we might come back and formulate another hypothesis, and even our hypothesis may lead us back to questions, and additional things that we need to discover. So what I'm getting at is the scientific method is not this unidirectional, we start with a question, we end with an answer approach always. Oftentimes there is rethinking, there's backtracking, there's rerouting that leads us down different paths, and it's still just as valid as this approach that we've been doing since the 5th grade. So just a little bit more on that. When we're thinking about hypotheses, we always want to make sure that hypotheses are testable. So that is a key word that you're going to hear over and over again when talking about formulating hypotheses. We have to make sure that our hypotheses are testable, and then we have to design our experiments that actually test what it is we're asking, what the question is. So, and sometimes our interpretation may lead us to either support that hypothesis or it might contradict our hypothesis, and we kind of go in this circular path, and that's okay. That's still a valid way to do science and research. So here's a good example of a problem. So my problem is my flashlight doesn't work. My question then becomes, why doesn't my flashlight work? And then I might have an option. So I've got two hypotheses here. Number one, my batteries are dead, or number two, my bulb is burnt out. Both of these are testable hypotheses. So if we take the pathway on the left for a second and think about batteries are dead, so we can predict that replacing the batteries will solve the problem and my flashlight will work. So we do our test, that's our experiment, we replace the batteries. We look at our results and we see the flashlight still doesn't work. So our hypothesis is contradicted or not supported. You never say it's proven or disproven. We would say supported or not supported. And in this case, this hypothesis was not supported. So we go back, right? We don't just, that's not the end of it. We still need our flashlight to work. So we kind of circle back to our question and circle back to hypothesis number two, that the bulb is burnt out. So we predict that replacing the bulb will fix the problem. We replace the bulb and the flashlight works. So this hypothesis was supported. So you can see that this was sort of a circular approach to science that brought us to our result. Now, remember I told you that we had to always think about our hypotheses in terms of being testable and falsifiable. That's another good word. So it has to be, we have to be able to falsify our hypothesis. It can't be something that is always going to be true. So one example that would have been a really bad hypothesis for our flashlight example would be that ghosts fooled with the flashlight and that's why it doesn't work. So we can't test that. That's not something that we can test in a scientific setting. And so that's sort of outside our bounds or outside the realms of science, and that's not a valid, testable, or falsifiable hypothesis. We could neither support nor not support that sort of hypothesis. So that's not a good hypothesis for us to think about in terms of scientific research. So some of those other things that we talked about in terms of the scientific method was with exploration and discovery. So this is where we're seeking to answer questions. We're observing nature. We're exploring the literature. We want to delve in and sort of find out what it is that interests us. When we talk about community analysis and feedback, so we're getting feedback on our research from peers. We are making sure that experiments are able to be reproduced and replicated. We're discussing with colleagues, we're publishing things, we're building new theories. All of these things fit into that feedback category and have a really important role in the research that we do. And then we think about what are the benefits and the outcomes of our research. So what societal issues are we addressing? Are we informing policy? Are we solving everyday problems? Are we simply satisfying our curiosity, which is very valid and a very important approach or reason to do scientific research. So all of these things fit into the quote-unquote scientific method and play a really important role in how we do research. So you're going to hear a lot more about this in lab when you start working on your independent research projects. And I really hope that during your career here at Marymount that you will choose to get involved in some research and do some independent research. I think it is an invaluable piece of your undergraduate education. And the science faculty here at Marymount have a very robust research program, and we love to see freshmen and sophomores get involved in that research. So don't think that just because you're not a junior or senior that you can't do it. We are going to be holding an open house pretty soon with all the labs open, so you can go in and talk to the professors and see what kind of research they're doing and think about how you can use these approaches to doing scientific research on your own in an independent way. So I will see you in class.